Return to room temperature. First, the room temperature must be maintained at around 25 degrees Celsius. Wait for it to stabilize and remove the test kit and the treated samples from the refrigerator. Allow the test kit to sit indoors for 30 minutes or more, allowing it to reach the same temperature as the room. This step is also referred to as balance of test kits. After the kit has reached room temperature, remove the individual components of the kit with their labels facing towards you for easy access. Open the required number of enzyme-labeled wells, and seal the remaining wells in the sealed bag. Place this bag inside the foil bag along with the desiccant. Then, put the foil bag back into the empty box, seal the box using a waterproof bag, and return them to the refrigerator immediately at 2 to 8 degrees Celsius. Plate setup. This process involves assigning numbers to samples and standards on wells. Each sample or standard is set in duplicate, meaning each into separate wells. The purpose of this step is to reduce experimental errors and correct operational mistakes. Additionally, it's important to note that parallel experiments must be conducted for all 6 standards each time. These are used for analysis and calculations. Parallel wells should be set in adjacent positions, either in rows or columns. It is recommended to arrange the plate in a vertical column format. The following is a demonstration of arranging them in a horizontal row. Sample reaction. Before actual operation, it's important to familiarize yourself with the procedures and key points. As per the instructions, the reaction involves adding the corresponding reagents into pre-numbered wells in the specified order. It can be said that an enzyme-linked immunosorbent assay is composed of the process of adding various reagents, making this step crucial to perform correctly. Repeated practice is essential to become familiar with this process. The key points for this operation are as follows. 1. Begin by adjusting the micropipette to the specified scale. 2. When picking up reagents, be sure to check if the labels are accurate. Open the bottle cap with the opening facing upwards to avoid contamination. 3. When using a micropipette, ensure that the pipette tip is inserted below the liquid surface in the reagent bottle to prevent the aspiration of air bubbles or inadequate liquid volume. 4. When dispensing reagents into the wells, do so swiftly and avoid touching the bottom of the wells or the well walls. It's best to dispense the liquid above the wells and avoid pipetting up and down after adding the liquid. 5. After adding each reagent, remember to change the pipette tip to avoid cross-contamination. You don't need to change the tip for the same reagent. Following the sequence established earlier for the plate setup, follow the steps and requirements outlined in the instruction manual. We will now proceed with the addition reaction demonstration. According to the instructions, we add the standard first. First, adjust the micropipette to 50 microliters. Attach a pipette tip. Pick up the standard, unscrew the bottle cap and place it on the table with the cap facing up. Immerse the micropipette below the liquid surface. Aspirate the liquid, remove it, suspend it directly above the well, and dispense the liquid. Change the pipette tips. It's important to note that you should change to a new pipette tip every time you switch to a different liquid. Continue in the same manner. After adding the standard, proceed to add the sample. Once the sample is added, follow by adding the enzyme conjugate. Finally, complete the process with the antibody solution. After adding the reagents as per the instruction, cover the plate with the adhesive membrane and mix thoroughly using a microplate shaker. Incubation. This process requires a specific temperature and should be conducted under conditions protected from light. You can choose from incubation equipment such as a constant temperature incubator or a water bath. As long as the temperature requirements are met, you can choose either one. Ensure the incubation equipment is preset to the correct temperature as per the instructions, and cover any observation window with opaque material to prevent light exposure. Place the plate into the pre-adjusted incubation device. Washing. This step is a crucial part of the entire experiment. During the washing process, please pay attention to the following 3 points. 1. The washing solution must be prepared in advance. 2. Ensure wells are fully filled with the working wash buffer. Consider using a multi-channel pipette for faster filling and better washing efficiency. Alternatively, an automated plate washer can ensure thorough washing. 3. In the final step, make sure to completely remove the liquid from the wells during the tapping process. If there are any bubbles, use a clean pipette tip to puncture them, as they can otherwise affect the subsequent experiments. After a 30-minute incubation period, remove the plate from the incubator. Gently lift the adhesive membrane. Support the wells with your fingers, and discard the reaction liquid into a designated waste container. Then, use a multi-channel pipette to aspirate the working wash buffer. 350 microliters per well, ensuring that the wells are fully filled. After allowing it to stand for 30 seconds, discard liquid in the wells. And then repeat the previous steps for a total of 5 wash cycles. After the final washing, pat dry with an absorbent paper. Color development. Following the instructions in the manual, first add 50 microliters of the substrate reagent A. And then add 50 microliters of the substrate reagent B. Ensure to change pipette tips when switching between different liquids. Cover with the adhesive membrane. Gently shake for 5 seconds using a microplate shaker, and incubate at 25 degrees Celsius in a light-shielded environment for 15 minutes for color development. If the blue color appears too light, you can extend the incubation time as needed. Add the stop solution. Then, add 50 microliters of stop solution to each well, gently shake using a microplate shaker, and terminate the reaction. ELISA reading. The ELISA reading is the process of presenting the entire test results through data interpretation. When taking readings, 2 points should be noted. 1. The microplate reader should be powered on 30 minutes in advance, and the wavelengths should be configured, with the primary wavelength set to 450 nanometers and the secondary wavelength set to 630 nanometers. If the microplate reader has only a single wavelength, select 450 nanometers for measurements. 2. The ELISA reading should be performed immediately and completed within 10 minutes after adding the stop solution. After completing the reading, the reaction solution should be disposed of in a designated waste container to prevent environmental contamination. During the experiment, always remember to wear lab coats, gloves, masks, and more. Avoid direct contact with reagents using your bare hands.